Okay, so there's going to be a short section of notes today, and you're going to kind of write out the calculations on your own that I walk you through, and then you're going to submit those for extra credit before you work on your problem set for today. It's just practice on different kinds of monohybrid crosses. So for today's notes, we're going to talk about Mendel's laws and his theories. And remember that he was the one who worked with pea plants, and he was using their characteristics and the fact that they are quick to grow to kind of go through and think about how traits are inherited and the patterns that they're inherited in. So before 1860, the thing that was most common was called the blending theory of inheritance. And it means that when parents mate and produce offspring, there's a mixed or intermediate combination of the parents' traits in the offspring. It's not like black and white. So if a black colored cat mated with a white colored cat, then the offspring would be gray. And that's what used to be the idea. And so Mendel crossed peas that had green pods with peas that had yellow pods. And so let me know, what do you think the color of offspring are expected if blending is true? Right. So the answer would have been a yellow-green color, right? So in between yellow and green, all of the offspring would have been this mixed color. And we all know that that's not really true, because when he actually crossed the F1 peas together of the progeny and the offspring, even though they were both green, right, he was like, first of all, they didn't blend because they were all green offspring. The progeny of blending green and green actually didn't just produce more greens. They actually produced the F2 progeny in a 428 green peas to a 152 yellow peas ratio. So what is this ratio? You can do that simply in your calculator. So 428 divided by 152 is about 2.8, which means that the ratio of green to yellow, if you round it to the largest numbers, is 3 green to 1 yellow, right? 2.8 is about 3 if you round up. So what do these numbers show? Well, they show what we were originally talking about when you crossed a heterozygous with a homozygous recessive, and, if you're crossing two heterozygous from the F1, right? Over here, we know that the plant is homozygous dominant and the other plant is homozygous recessive. So all of their offspring actually ended up being this F1 progeny. So they're all green. So they all show the dominant phenotype, but they're actually all heterozygous because they got one dominant from one parent and a one recessive from the other parent. Now, when you have the dominant showing phenotype, but actual heterozygous genotype, the offspring is always a 3-to-1 ratio of phenotype of dominant phenotype to recessive phenotype. But we know the genotype is actually a 1 to 2-to-1 ratio of one homozygous dominant, two heterozygous, and one homozygous recessive. So these numbers actually show that there is no blending and that it's actually a dominant recessive allele relationship for the gene that covers the pea pod color. So Mendel got the same results with 6 other characteristics. So I'm going to ask that you calculate out the ratios of dominant and recessive for stem length, pod shape, seed color, flower position, flower color, and pod color. So I want you to calculate the phenotypic ratios that he saw. For example, 787 divided by 277. That gives you a number, which is the number that belongs to the dominant phenotype. You'll round it to the nearest whole number, and then the recessive should always be 1, since that is the denominator that you're dividing by. So after you figure out those ratios, I want you to kind of explain what these numbers show. It's kind of related how I explained back here for the question on what do these numbers show after you find the ratio. Right? I kind of want you to think about, does blending really exist? Did Mendel support or disprove the blending theory of inheritance? Number 2, using this data, figure out Mendel's law of segregation. So you can talk about what happens to alleles when meiosis is occurring. Do they stay together or do they separate? Are there rules to how they stay together or separate? Or is it random? So you tell me. If you're really, really struggling, you can Google it if you need to and give me a personally worded version of what you find. So here is the actual law of segregation. Each individual has a pair of factors or alleles for every trait. So there's 2 alleles for each trait. You either have 2 of the same or 2 different ones. And the reason why you can have 2 different ones is because usually there's a dominant and a recessive, or there's 2 versions. Some genes actually have multiple versions, so there's more than 2. The factors or alleles segregate or separate during gamete formation, which is when you're forming sperm and egg. So these alleles go their own ways, right? When the homologous pair is separating, these alleles separate on their own. Each gamete contains only one factor or one allele from each pair of factors, right? So if you have a homologous pair and they separate, that means that you're having half the amount of chromosomes in each gamete, each sperm or egg, and therefore you can only get one of the 2 alleles that the parent originally has. And the last part of the law of segregation is that fertilization gives the offspring 2 factors for each trait. So the sperm contains one allele and the egg contains the other allele that are going to work together in the offspring to produce a specific genotype and a specific phenotype. Okay, that's when you go from 1 to 1 plus 1. Fertilization creates, again, 2 factors for each allele. That way you can actually have the relationship in the genotype of recessive, dominant, or codominant, or incomplete dominance. Okay, so these are actually Mendel's laws. I would love it if you could have them written down, and then I will check it in your notes. Again, after this, you have your problem set 1, where you're going to work on monohybrid crosses, and later this week, we're going to touch on dihybrid crosses.